Okay, we're going to use the example of sickle cell anemia to talk about human diseases which are caused by mutations. So it's a really good example of how a change in a single base in the gene can have a massive effect on the health of the individual. Right, so sickle cell anemia then, okay, it's caused by a mutation to the beta chain in hemoglobin. Okay, so here's hemoglobin. Hemoglobin is made up of 4 polypeptides, 2 identical alphas and 2 identical betas. Beta chains, each one has a heme group, and each one of those subunits binds 1 molecule of oxygen. If you have the normal gene, the normal gene for beta globin I'll call capital S, and the mutated gene we'll call little s. Okay. We'll talk about what that mutation is in a minute, but sickle cell anemia shows kind of it behaves like a recessive disorder mostly. Right. So if you have this this phenotype here, 2 good copies, 2 normal copies of the beta gene, then that person is healthy. If you have 2 mutated copies, this one here, this person has sickle cell anemia, sickle cell disease. Life expectancy is massively reduced. Frequently people die before their 30s. And if they have this, they're heterozygous, then they're not completely healthy. So you could call it kind of like an example of codominance, but they're more or less, they don't experience that many problems. So we'll just say healthy there. Okay. Now, well, let's see what happens in sickle cell disease. We'll start off with the mutation. Okay, so the beta globin gene, in the normal version of it, there's several hundred amino acids long, and I think it's the 6th or 7th one along. The 6th or 7th codon in the healthy gene is GAG. So that's in the healthy gene. Here's in the sickle cell gene. Well, that one of those codons is changed, so you get GTG. Now that causes the amino acid which it codes for to change. In the normal version, it's glutamine, glutamic acid, glutamate, GLU, whereas and that gets changed to valine in the sickle cell. Now just to show you here that there are 2 codons for glutamate. There's GAG and there's also GAA. So if the mutation had occurred not in the middle base, but on that end one, and it changed to an A, then although there'd be mutation to the gene, it had actually no effect on the phenotype whatsoever, because it'd be the same amino acid. Right. Let's see why just changing 1 amino acid in 300 or 400 has such a large effect. Okay, so if you just draw the structures of those 2 amino acids, the R groups, glutamate, the R group is CH2, and then it has this charge group at the end, and this makes it pretty hydrophilic. Whereas if it gets replaced by a valine, that is not hydrophilic. You get a CH and 2 methyl groups stuck to the carbon there. That's very hydrophobic. And that makes all the difference. So this is in the sickle cell gene. So this is the mutated version. And that has the effect. Normally this hydrophobic R chain is on the outside of so there's your beta globin gene, and it will stick somewhere on the outside here, and it will interact with the aqueous environment of the cell. When you've got the valine there instead, this one, it can't do that. So what tends to happen is that one hemoglobin molecule forms non-polar interactions with another hemoglobin molecule. So the hemoglobin molecules all stick together. They stick together and they form clumps or aggregates, and that actually distorts the shape of the red blood cell. Okay, so it affects the cytoskeleton of the red blood cell. So red blood cells, of course, are normally red blood cells are biconcave disc. And what happens if you have the sickle cell hemoglobin, too much of the sickle cell hemoglobin there, the cell goes from being this biconcave disc to being a sickle shape. And these can be seen in the patient's microscope, patient's blood under the microscope. The red blood cells are this, certainly all of them, at some of the sickle shape. And this causes problems because these tend to stick together and form clots. And blood clotting occurs, and they form blockages in the capillaries. So you can have blockage in the capillaries almost anywhere in the body, but very often, if they form a blockage in the brain, then this can lead to a stroke. So sickle cell sufferers tend to suffer from strokes at an early age. They block the blood vessels in the heart. They can have heart attacks, and also it causes a lot of damage to the kidneys as well. In the nephrons of the kidneys, these aggregates stop the blood being filtered properly. So it has very large effects on a lot of different organs in the body and leads to an early death. Okay, so let's talk about how we know now then that, right, this single base getting changed from A to T, that causes everything. But how was this discovered? Let's talk about how it was discovered then. Right, so this mutation is very prevalent in Africans in sub-Saharan Africa. It's very common mutation. I think it's 4% of the population carry the mutation, and in African populations all over the world, they suffer from it as well. Right. So it was discovered about the late 19th century. People obviously knew this disease existed. The late 19th century, somebody actually looked at the red blood cells under a microscope and they observed sickling. And they didn't know what caused it, but they could see these cells were causing sickling, and they rapidly began to understand that they were going to cause clots, which would cause problems. Right. Then in, I think, it's quite late on, I think it's in the 70s, someone, they realized the problem was with the hemoglobin, and they narrowed it down to the beta globin gene, and so they partially digested the beta globin with protease enzymes, and then they separated the fragments by electrophoresis. So you get a characteristic series of fragments in healthy people, and you get a very similar series of fragments with people with sickle cell. Most of them are the same, so one fragment was different. It moved at different rates during the electrophoresis, showing a different chemical composition. So at that point, due to the work of Sanger, proteins could be sequenced, small proteins could be sequenced. So the amino acid sequence of the fragment was determined. And that's where they spotted the glutamic acid residue was swapped for a valine residue. So they'd put down at that point then, so these global changes to red blood cells could then they're pretty confident it was caused by this change from the amino acid glutamate to valine. At that stage, in the 70s, DNA technology was obviously nowhere near as advanced as it is nowadays. And it was not until about 1980 that they managed to clone the gene for beta globin. They cloned it and they sequenced it. Right, how did they manage to do that? Well, they made a cDNA library. Okay, so cDNA library from red blood cell precursors. Red blood cells and stuff, you can't make it. Remember what cDNA is basically a copy of the mRNA in the cell. Of course, red blood cells don't have any mRNA or ribosomes or nucleus, but their precursors do, and that's where they're making all the hemoglobin. So they red cell precursors made a cDNA library. They identified the one clone which was carrying the beta globin mRNA. And how did they find that one? Well, they worked backwards, and they got the protein sequence, and from that they deduced the base sequence. They knew the genetic code at that time, and they made an oligonucleotide probe, and that will hybridize hydrogen bond to the DNA for the hemoglobin, for the beta hemoglobin gene. They identified that. So once you've identified the clone in the cDNA library that contains this beta globin gene, you can get lots of it, and then they could sequence it. In the 80s, it was a much slower process than it is nowadays. Not automated. This was way before the Human Genome Project, but they did sequence it, and they sequenced it from people who are suffering from hemoglobin, and they found out that sickle cell sufferers have that mutation for the, what was it? It was the GAG, gets converted to G, just the T, isn't it? The GTG. That's all it. Add in the English this timing, that causes all the problems. So you may think, well, if this is this sickle cell gene is obviously, so if you get a double dose of the gene, it's very harmful. The person won't live to adulthood. Very unlikely that they would be able to pass it on to their offspring, because they're just not, they'd be infertile, they're not well enough. So why hasn't it been sort of eliminated by natural selection? Well, it was known that sickle cell tended to come from areas where there's a high level of malaria, when malaria was endemic. And it turns out that when you are heterozygous, that gives you some form of resistance to malarial infection. And so when you have it in a single copy of it, it gives people a selective advantage if they live in an area of malaria. And remember that in countries where malaria is endemic, you know, quite a significant percentage of infants don't survive past their first birthday because they get malaria infections which kill them. So it's a very big selective advantage if you can resist malaria. How does that work? Well, the malaria parasite is eukaryotic. It's not a bacteria. It's a protozoan, and it's got quite a complicated life cycle. It goes in the salivary glands and mosquitoes and stuff. But in humans, it tends to go inside red blood cells. Okay, and it uses the red blood cell as food, and it multiplies. And what that will do, it will lower the oxygen concentration in the red blood cell because the parasite is respiring. And at low oxygen concentrations, the hemoglobin, when it's deoxygenated, they tend to form these clumps more. So at low oxygen concentration, hemoglobin causes these clumps, and the cell becomes sickle shaped. Right, when this passes through the spleen, this is removed by the spleen. So you can see that it's almost like a filter for removing any red blood cells that contain malaria parasites. And again, even if they've only got one bad copy of the gene. With the malaria parasite in the red blood cell, the oxygen concentration drops. It is enough to make it sickle, and that will be removed, so you don't get the malaria parasite dividing. Okay, so that's probably one of the best examples of a disease which has been caused by a single mutation. Just before we finish, just say that there we have amino acids we have nucleotides come in groups of 3. So we had our G, A, G. Right now, it's called a point mutation when you swap one base for another. If you have something like an insertion, so certainly if we put an extra base in there, so we inserted another one, that tends to have a more catastrophic effect on the protein, because what that will do is it will shift the codon is being read there, the frame is gone, it's called a frame shift mutation. And that will it means that after the mutation, every single amino acid will be wrong. So you're going to get a completely non-functional protein, so you won't make any protein at all. With point mutations, you usually do get the protein. And we said point mutations may lead to a change in amino acid. It might not, even if it changes the base. Sometimes even if it does change the amino acid, it changes it to one where it's not in a crucial part of the protein molecule, it won't make any difference to the protein's function. So they'll be kind of silent mutations. Although there is a mutation, yet the amino acid changes, it doesn't have any big effect on the organism.